Hello friends, welcome to the Pathology Insights. Today in this video I'll be discussing about amyloidosis, and only I'll be discussing about what is the definition of amyloidosis and what are the physical and the chemical properties of the amyloid protein. Remaining classification and morphology we'll be discussing in my next video. So coming to the definition, amyloidosis is a heterogeneous group of the disorder which can be acquired or the hereditary, and it is characterized by predominantly extracellular deposition of abnormal fibrillar protein in various tissues. Now this protein, what I'm telling is predominantly extracellular. In some cases it can be intracellular also, but predominantly it is extracellular. So in the initial stages, amyloidosis, it doesn't produce any signs and symptoms. Patient will be asymptomatic. But as the deposition in between the cells increases, it encroaches upon the cells and the cells undergo the pressure atrophy. So when the atrophic changes occurs in the cells, obviously the tissue, the function of the tissue is affected, and this leads to the tissue damage and the functional compromise of the tissue wherever we have this deposition of the amyloid fibrils. So as I told you, amyloid fibrillar protein is because of the aggregation of this misfolded protein. So normal mechanism of the body, how it removes of this misfolded protein, we'll see here. So normally the proteins are produced by the ribosomes when they come in contact with the endoplasmic reticulum, and the proteins which are produced, they will be folded, and with the help of the chaperones they will be directed towards the organelles where they are required, or they will be transported to the Golgi apparatus. This is when the protein is properly folded. If the protein is misfolded, it cannot be used by the organelles. So this misfolded protein will be transported out into the cytoplasm. And in the cytoplasm, the ubiquitin molecules, they combine with this misfolded protein and direct this misfolded protein into the proteosomes. So in the proteosomes, these misfolded proteins, they undergo degradation. So this we call it as a ubiquitin proteasome mechanism, which is useful for removing the misfolded proteins. This is a normal mechanism. Now we have the aggregation of the misfolded proteins if this mechanism is not functioning normally, or if we have increased production of this misfolded proteins. So if we have increased production of the misfolded proteins, they aggregate, they polymerize, and along with that they also accumulate certain other molecules also, like proteoglycans, glycosaminoglycans, and serum amyloid P proteins also, and also they undergo certain conformational change in the structure and they form amyloid fibrils. So this is how the amyloid fibrils are formed from the misfolded proteins. So I told you after the polymerization, it should undergo some conformational change in the structure, and that is they form cross beta pleated sheet structure. Now once they form this cross beta pleated sheet structure, these proteins, they become hydrophobic, they are non-functional, insoluble, and even they are resistant to the degradation. So that's why they can't be degraded, and then they start accumulating. So this is the cross beta pleated sheet structure which we see in this amyloid fibrils. Now when we see the history, why the term amyloidosis is used for them is, this is because of their staining characteristic, which is similar to the starch. So the starch, when we apply the iodine on the starch, it gives a brown color. So these organs which had the deposition of the amyloid, when it was stained by the iodine, it used to give the brown color. So hence they thought the material is starch-like material. Now why the properties has come like a starch is, there was abundant charged sugar groups in this adsorbed proteins. So because of this sugar groups, it has the characteristics, similar staining characteristics as that of the starch. So initially, because it has a staining characteristics of the starch, which was in the Latin, the cellulose or starch-like material is called as amylum, and in the Greek it's called as amylon. So because the deposited material is having the similar characteristics, from these two terms they have coined the term as amyloid. Rokitansky was the first person who has described the deposition of this starch-like material in 1842. Later on, the term amyloidosis was given by the Rudolf Virchow in 1854. But still here they were thinking that it is the starch-like material only, based on the color produced by the staining with the crude iodine. In 1859, Friedrich and the Kekule were the persons who recognized that this is not a cellulose or the starch-like material, but instead it is a protein which is deposited in the amyloidosis. This is about the history of the amyloidosis. Now coming to the physical nature of the amyloidosis, when we see under the electron microscopy, amyloid fibrils, we have different types of amyloid fibrils like AL, different types are there. Irrespective of the type of the amyloid fibrils, all the type of the amyloid fibrils, they are continuous non-branching fibrils which have a diameter of approximately 7.5 to 10 nm. This is how they appear in the electron microscopy. Now the cross beta, as I told you, the structure is cross beta plated sheet. So this cross beta plated sheet structure on X-ray crystallography and infrared spectroscopy, it gives cross beta diffraction. So this structure was cross beta plated sheet structure was confirmed by the X-ray crystallography, which gives a cross beta diffraction. And also this cross beta plated sheet is responsible for the special stains what we use, like the Concorde stain is specifically used to demonstrate the amyloid. Because of this cross beta plated sheet, the Concorde stain it gives positive to the amyloid. By using the amyloid stain in the light microscopy, amyloid deposited material, it gives a red pink color. But the same thing when we see under the polarized microscopy, we have the apple green birefringence. If you see here, this the light green color, the shining thing, what we call it as an apple green birefringence, which we see in the polarized microscopy, same on the Concorde staining. Concorde staining in the light microscopy, it gives red pink color, whereas similar if we see in the polarized microscopy, it gives apple green birefringence. That was about the physical properties. Now when we see the biochemical properties of the amyloid, we have nearly 20 different proteins, but here we will be discussing only about a few important ones here. So we have 3 major forms and we have 5 minor forms here. So under the major forms we have amyloid light chain, amyloid associated protein and beta amyloid protein. If you see the terminology, A stands for amyloid, L stands for the light chain, and here also A stands for amyloid, A stands for amyloid associated protein. Similarly, A for amyloid and beta for the beta amyloid protein. That means the initial letter, it stands for the amyloid, and the second letter, it stands for the protein which is deposited in this type of the amyloidosis. And then in the minor forms we have transthyretin protein which is deposited, we have beta 2 microglobulin, calcitonin, islet amyloid peptide, and we have atrial natriuretic factor. Now we will see the individual one. So in that one of the major form was amyloid light chain. For this the precursor protein is immunoglobulin light chain. Now if you remember the structure of the immunoglobulin, it has a heavy chain and it has a light chain. So both heavy and the light chain have a constant component and a variable component. So in this amyloidosis we have either this terminal fragment of the light chain or we have the complete light chain which is deposited. And this condition usually we see in the plasma cell tumors, where the plasma cells will be producing excess of the light chains. So these light chains, they aggregate, they form the oligomers or the intermediates, then they undergo certain conformational change and they form the amyloid fibril. So amyloid light chain type of the deposits we see in the plasma cell tumors. Next one is about the amyloid associated protein. Now this we see in the chronic inflammatory condition. So in the chronic inflammatory condition, the chronic inflammatory cells we have are the macrophages and the monocytes. Now these inflammatory cells, they will be producing number of the cytokines. In that, the important one are interleukin 1 and the 6, which will be stimulating the hepatocytes. Now these hepatocytes, under the influence of interleukin 1 and the 6, they produce serum amyloid associated protein. Now further, this serum amyloid associated protein, it undergoes the proteolysis to produce amyloid associated protein. So the conditions where we see is in the chronic inflammatory condition, and the precursor protein is serum amyloid associated protein, which is a non-immunoglobulin protein. So that is about AA protein. Then the third major form is beta amyloid protein. Now for this, the precursor protein is a transmembrane glycoprotein in the neurons and the astrocytes, which is called as amyloid associated protein. You see, this is the membrane and this is this transmembrane glycoprotein, which is called as amyloid associated protein. Now normally this amyloid associated protein, it undergoes proteolysis by alpha secretase and the gamma secretase and forms the soluble components, which doesn't accumulate normally. But if it is acted upon by the beta secretase and the gamma secretase, then they break at the point where they form the monomers, that is A beta monomers. These monomers, they aggregate and form A beta oligomers. Later on, these oligomers accumulate, undergo certain conformational change, and they form the amyloid fibrils. Now these amyloid fibrils, they get deposited in the brain. They form the plaques, then they form the neurofibrillary tangles, and they get deposited around the blood vessels also. This we see in the condition called Alzheimer's disease. Now that three were the major forms. Now we see the amyloid, the minor forms, which is amyloid transthyretin. Now here the protein which is deposited is transthyretin, which is a normal protein that is useful for transporting thyroxine and retinol. So in the transthyretin, trans stands for transporting, thyretin, Thi stands for thyroxine, retin stands for retinol. So this is a protein which transports thyroxine and the retinol. In this we have two forms, one form where the normal TTR is deposited and another form where we have mutated TTR which is deposited. So this TTR is normally produced by the liver, and the TTR normally it has a tetrameric structures. So when these tetrameric structures it is dissociated, we get the monomeric forms. So these monomeric misfolded forms, they aggregate and they form the amyloid fibrils. So in the long conditions, chronic conditions, these amyloid fibrils, they get deposited in different organs. We call this condition as senile systemic amyloidosis, but in this condition the most commonly affected organ is heart. Now here this transthyretin is a normal TTR, whereas there is another condition where we have production of mutated TTR which cannot be disintegrated. So these mutated TTRs, they aggregate and they form the amyloid fibrils. Now these amyloid fibrils, they get deposited in the peripheral nerves and in the autonomic nervous system, leading to familial amyloid polyneuropathies. So now it's clear, if it is a normal TTR, a fragment of the normal TTR which is getting deposited, it leads to senile systemic amyloidosis. If it is a mutated TTR which is getting deposited, that leads to familial amyloid polyneuropathies. The another minor form is amyloid beta 2 microglobulin. Now this beta 2 microglobulin is derived from MHC class 1 molecule, major histocompatibility complex class 1 molecule. Now this molecule is present in the antigen presenting cell on the membrane, and this molecule has four components: alpha 1, alpha 2, alpha 3 and beta 2 microglobulin. This is useful for stabilizing this molecule, And when this is released into the circulation during the inflammation, it undergoes the proteolysis, and the soluble component of it, which is formed by the proteolysis, is useful for chemotaxis and bacterial killing. And sometimes the proteolysis leads to the oligomers, which leads to the formation of the amyloid fibrils. Now that is one cause. And another thing is, in the acidic pH, whenever there is an acidic pH, this beta 2 microglobulin can form the aggregates leading to the amyloid fibrils. We see this type of the amyloidosis in the conditions where the patient has undergone long-term dialysis. Because when the patient has undergone the dialysis, during the filtration, this is the protein which cannot be filtered out. So this remains, is beta 2 microglobulin, it remains in the blood itself, and hence the serum levels of this proteins increases in the patients who are undergoing the dialysis. Now once the levels are increasing, they will be getting deposited in the different organs. But nowadays we are having recent filters which are able to remove it also, so hence the incidence is getting reduced. But previously, the patients who are having a chronic renal failure and who are undergoing the dialysis were having the complication of this beta 2 microglobulin deposition in the different organs. Other rare forms we have is the calcitonin deposition, which is produced by the tumor cells in the medullary carcinoma thyroid. We have islet amyloid peptide, which is deposited in by the pancreatic islet cell tumors and in the type 2 diabetes. Atrial natriuretic factor is one which is deposited in the atria. Serum amyloid P component, apolipoprotein E, and sulfated glycosaminoglycans can also be deposited, which can lead to the amyloidosis. These are the rare forms. Now when we see the summary of the amyloidosis, first we have to remember the definition, that amyloidosis is accumulation of misfolded protein fibrils, which is mostly extracellular. And when we see the structures, they are continuous non-branching fibrils having a diameter of 7.5 to 10 nm, and they have a cross beta pleated sheet structure, which is confirmed by X-ray crystallography, which produces cross beta diffractions. So when we see biochemical forms, we have major forms and the minor forms. In the major forms, we have amyloid light chain, which we see in the plasma cell tumors, and the precursor protein is immunoglobulin light chain for this. Second one is amyloid associated protein, which we see in the chronic inflammation. Precursor protein for this is serum amyloid associated protein produced by the liver. Then the third one is beta amyloid protein, which we see the deposition of this in the Alzheimer's disease in the brain, and the precursor protein for this is transmembrane glycoprotein. Now the minor forms, when we see, we have transthyretin. Now in this we have mutated form and the normal transthyretin also. The mutated form of the transthyretin, this leads to familial amyloid polyneuropathies, whereas the normal TTR, it leads to senile systemic amyloidosis, in which heart is the organ which is commonly affected. And beta 2 microglobulin, in this condition we have we see in the dialysis patients. Precursor molecule for this is MHC class 1 molecule from which it is derived. And calcitonin, which is produced in the medullary carcinoma thyroid by tumor cells. Islet amyloid peptide, it is seen in the type 2 diabetes. And atrial natriuretic factor, we see the deposition in the atria. So these are the minor forms and these are the major forms. So that finishes about the physical and the chemical nature of the amyloid fibrils. In my next video, I'll be discussing about the classification and the morphology. Thank you, friends. Thank you for listening patiently.